In this video, we're going to look at a very brief overview of metabolism, specifically focusing on catabolism. Right, so metabolism is going to be the sum of all chemical reactions that take place in an organism. So this means it includes catabolism, which is the breakdown of large molecules, and it includes anabolism, which is going to be the reverse process, which is the synthesis. So here I'm showing you a depiction of each. In catabolism, we're going to start from some very large molecules, and then we're going to break those down in a series of reactions in order to generate some smaller molecules and some energy shown here as ATP. So we're going to look more at this molecule here, this ATP, in a separate video. But right now, ATP is going to be what we mean by producing energy. It's producing these ATP molecules. So that is catabolism. In anabolism, we're going to do the reverse process, meaning we're going to start from smaller molecules, combine that with some energy, because this process requires energy in order to create these much larger molecules. So in metabolism, you're going to have both processes happening. Sometimes you're going to need to break things down to produce energy, and other times you're going to need to store large molecules, and so you're going to synthesize these together into larger molecules. In metabolism, there are two different types of pathways that we often talk about. So the process of metabolism is a long series of consecutive reactions, and this is what we call a metabolic pathway. These pathways can either be linear or they can be cyclic. So what I'm showing you here is these compounds A, B, C, and D. These are all different organic molecules. In order to go from compound A to compound B, there is an enzyme that will help catalyze this reaction. I will call this enzyme 1. Then there will be another enzyme here. I will call this enzyme 2. That is going to help convert the organic molecule from B to C, and then C to D. Here I'm showing you an example of a linear metabolic pathway, meaning compound A is converted all the way down to compound D. This is in contrast with a cyclic metabolic pathway, where again, your compounds are converted from A to B again with an enzyme, but the difference here is that at the very end, you are going to produce compound D, which is then converted back to compound A. And this is what makes it cyclic. It's able to reproduce the starting material. What these two have in common is that in order for these pathways to happen, you do need enzymes. Independent of whether you're doing catabolism or metabolism, your enzymes will help speed up the reactions. The last thing to talk about before we look at some more details about catabolism is where these things are happening. So most of the energy production, again, what I mean by this is production of your ATP molecules. Most of the reactions involved in producing this are going to occur in the mitochondria. So let's briefly look at the anatomy of a mitochondria. So here I'm showing you a eukaryotic cell, so similar to a mammalian cell, a cell that we have in our bodies. And this image is showing you the nucleus here, which contains all of your DNA, and then your cytoplasm, which contains all of these different organelles. The organelle that we are interested in is this mitochondria. So if we zoom into the mitochondria, we're going to see that it has two membranes. It has an outer membrane, shown here in brown, and it has what we call an inner membrane, shown here in yellow. Then there is a space in between the inner membrane and the outer membrane called the intermembrane space. And this space is actually going to be very important in the production of ATP, and we will look at that in much more detail in a separate video. Lastly, there is a matrix in your mitochondria. This is going to be the space that is inside your inner membrane. It's analogous to the cytoplasm inside a cell. This matrix here is just the region inside this double membrane organelle. We're going to see that some of the catabolic reactions we're going to look at will occur inside this matrix. All right, so now let's talk about catabolism, specifically looking at when you have large molecules being broken down into smaller ones with the objective to produce ATP, meaning to produce energy. So this is what we're interested in. Right? So there are 4 stages that we can organize in terms of how to organize catabolism. So in stage 1, we're going to look at what we call digestion. Then stage 2 will be formation of acetyl-CoA. Here, this is the molecule of interest. In stage 3, we're going to look at the citric acid cycle, often called TCA cycle. You will hear me use these two terms interchangeably. And then the last stage is going to be this electron transport chain, which I'm going to summarize by using ETC. All right, so we're going to briefly look at each stage, but stages 3 and 4, we are going to look at much more deeply in separate videos. The purpose of this video is just to give you a brief introduction to catabolism. All right, stage 1 is going to be digestion. This is where we are going to obtain our very large molecules. So the large molecules we're interested in are these biological macromolecules that we've already talked about, which is going to be the lipids, the carbohydrates, and the proteins. And here I'm showing you different sources for these very large molecules. So in stage 1, the purpose of digestion is to hydrolyze these biological molecules into their basic building blocks. Those basic building blocks are these fatty acids, the monosaccharides, and the amino acids. And the reason this has to happen is because these building blocks are the ones that are absorbed by your intestinal cells, and then that is what is transported into your circulatory system. So when we absorb food, they first need to be broken down in the digestion stage in order for them to be small enough to be taken into the intestinal cells and then into the circulatory system. Once they're in the circulatory system, they then can reach the destination tissue in which they will be needed. So let's look at these hydrolysis reactions just a little bit more closely. We have talked about these throughout the semester, and I just want to recall here what we mean when we say hydrolysis. And additionally, I want to talk about the enzymes that are involved in this process. So first, let's look at carbohydrates. Up here, I'm showing you the carbohydrate starch, and recall that starch is a polysaccharide. It's a very complex molecule. It's a very large molecule, and it's made up of glucose monomers, and they are linked together through this glycosidic linkage shown in red here. So it is this glycosidic linkage that is recognized by the enzyme amylase, written here, which is found in your saliva. So when you consume starch, the amylase will recognize this glycosidic bond here, and it will break it up in order to produce the individual glucose monomers or monosaccharides. Okay? It is going to be this species, this molecule, that is taken up by your intestinal cells. Next, we have proteins. Proteins are a large polymer of amino acids, as we've talked about before. And these amino acids are again joined by a peptide bond or an amide bond. Those two things are the same thing, which is shown right here in red. So when you consume protein, the enzyme pepsin, which is located in your stomach, is going to recognize this amide bond and it's going to cleave it. It'll cut it. And the consequence of this is that you will release the individual amino acids that are present in your protein. And again, it is these amino acids that are again taken up by your intestinal cells to enter the circulatory system. Lastly, we have our triacylglycerols or triglycerides. And again, this is going to be recognized by lipase. This is the enzyme that is going to cleave these ester bonds here in order to release the building blocks of triglycerides, which is going to be your glycerol molecules and your fatty acids. This lipase enzyme is found in your intestines, in the small intestine. All right. So that is stage 1. Again, the purpose of stage 1 is to take these very large complex molecules and break them down into its basic building blocks in order for them to be absorbed into the body and into the circulatory system so that they can reach the tissues where they need to be. So once they reach the tissues where they are needed, they're going to enter the cells that are in those tissues. Okay? So now in stage 2, what's going to happen is these linear metabolic reactions, which are shown here as fatty acid oxidation, glycolysis, and amino acid catabolism. Most of these will occur in the cytoplasm of the cell. So once these building blocks have reached their destination, they get taken into the cells in those tissues, and then most of these reactions are going to happen in the cytoplasm. So what is happening here is that these individual building blocks are being further broken down into one common molecule. That molecule is acetyl-CoA. And this is important because it allows 3 different building blocks that have very different structures to be all combined into one particular structure. And this is going to allow metabolism, in this specific case, catabolism, to be streamlined into just one pathway, one common pathway. With acetyl-CoA, we will look at this molecule a little bit more deeply, but right now, the important thing to note here is that the carbons in acetyl-CoA, so there's a carbon here and another carbon there. I'm showing you that over here in this type of formula, where the carbons in this acetyl-CoA are carbons that are coming from those monosaccharides, amino acids, and fatty acids. So it is these carbons that are being bound to this coenzyme A, which we are also going to talk about in more detail. Okay? But this acetyl-CoA molecule, again, those carbons here are coming from your lipids, your carbohydrates, and your proteins. And it's going to be this molecule that is going to enter the next stage, which is the citric acid cycle. So at this point, the acetyl-CoA is going to enter the citric acid cycle. And now these reactions, which is a circular metabolic pathway, these reactions are going to occur in the matrix. So the purpose of this cycle is going to be primarily to produce these reduced coenzymes. So when acetyl-CoA enters your citric acid cycle, there's going to be a series of reactions that are circular in nature. But this series of reactions, yes, it will produce some ATP, but the main point will be to produce these reduced coenzymes. And that's because it's going to be these coenzymes that enter the next stage. These coenzymes are specifically NADH and FADH2, and we will also talk more deeply about these in a separate video. All right, so now these reduced coenzymes are going to enter the electron transport chain. The electron transport chain is going to be located in the inner membrane. So stage 3 happens in the matrix in here. And now that you've produced these coenzymes, they are going to go to the electron transport chain, which is located in this yellow inner membrane here. Okay, this electron transport chain looks something like this, shown here on the left-hand side, where you have your membrane shown here with these blue spheres and the tails. This is your lipid bilayer. And then these yellow figures here are proteins. These are all different proteins, meaning they are also different enzymes that are located within this membrane. They are embedded in the membrane. And this NADH and FADH2 is going to be fed into this electron transport chain. There are going to be some reactions that will happen, but the ultimate goal here is going to be to produce ATP. Okay, because again, recall that this is our main goal here in catabolism, is to start from large molecules and produce these ATP molecules. So these ATP molecules are going to be produced within the mitochondria, specifically on this inner membrane of the mitochondria. That's where all the action is happening with the electron transport chain. Okay. So let's summarize. We're going to describe just the main features of the 4 stages of catabolism. And here we're focusing on the products of each stage because one will feed into the other. So in stage 1, that's digestion. Again, the point of this is to take large molecules and break them down to their basic building blocks. That's fatty acids, monosaccharides, and amino acids. In stage 2, the point of it is to form acetyl-CoA, meaning that each one of those basic building blocks is going to be converted into acetyl-CoA. Once you have that acetyl-CoA molecule, now you can go into stage 3, where that acetyl-CoA molecule will participate in a series of reactions in the circular pathway in order to produce your reduced coenzymes. This is the important part here of stage 3. Those reduced coenzymes will then be fed into what we call the electron transport chain, which is a series of many enzymes, but the point here is going to be to produce those ATP molecules at the end of this sequence. Okay. In a separate video, we are going to look at stages 3 and stages 4 in more detail, but first we're going to need to talk about what ATP is, its structure, and then we're also going to look at the structures of reduced coenzymes in order to then be able to dive more deeply into stages 3 and stage 4.